Chapter 7. Heredity and Cancer. This chapter explores how inherited genetic mutations contribute to cancer development. While most cancers are caused by environmental exposures, a significant percentage, about 10 to 20%, have a hereditary basis. Understanding hereditary cancer helps us recognize high-risk individuals and develop preventive strategies. Let's begin by reviewing the general patterns of genetic contribution to cancer. Approximately 80 to 90% of all cancers are attributed to environmental causes like diet, tobacco, or radiation. However, 10 to 20% of cancers result from inherited genetic mutations passed through families. These hereditary cancers are often linked to specific genes that are defective at birth. Individuals with these mutations are more susceptible to developing certain cancers, often at younger ages. Early detection and genetic counseling play a vital role in managing hereditary cancer risk. Genes exist in different forms called alleles, which may be dominant or recessive. In hereditary cancer, whether or not someone develops cancer can depend on a concept called penetrance. Complete penetrance means everyone with the mutation will develop the disease, while incomplete penetrance means some individuals will not. This variability can influence how cancer risk is inherited and expressed. Understanding penetrance helps genetic counselors assess risk more accurately. Retinoblastoma is a rare eye cancer that occurs mostly in young children. In familial cases, the RB gene mutation is inherited from a parent, while sporadic cases require two spontaneous mutations in the same retinal cell. Familial RB often affects both eyes and develops at an earlier age. In contrast, sporadic RB usually affects only one eye. This example clearly illustrates the difference between inherited and non-inherited cancer types. The RB gene is located on chromosome 13 and acts as a tumor suppressor. It regulates the cell cycle, specifically the transition from the G1 phase to the S phase. When RB is functioning, it prevents uncontrolled cell division. However, when mutated, this regulatory function is lost, leading to unchecked cell growth. RB mutations are involved not only in retinoblastoma, but also in several other cancers. The two-hit hypothesis explains how both alleles of a tumor suppressor gene must be inactivated to cause cancer. In familial RB, one defective allele is inherited and the second hit occurs later through mutation. In sporadic cases, both hits occur randomly in the same cell. This model helps explain why hereditary cancers often occur earlier and more frequently. It also underscores the importance of tumor suppressor gene function. The APC gene is another important tumor suppressor, especially in the context of colon cancer. Inherited mutations in APC cause Familial Adenomatous Polyposis, or FAP. Individuals with FAP develop hundreds of colon polyps during adolescence, greatly increasing their risk of colon cancer. APC normally regulates the WNT signaling pathway to control cell growth. Loss of APC function leads to uncontrolled cell proliferation in the colon lining. Li-Fraumeni Syndrome is a rare but serious hereditary cancer syndrome. It's caused by an inherited mutation in one copy of the p53 gene, a key regulator of apoptosis and DNA repair. This syndrome dramatically increases the risk of developing a wide range of cancers, including breast cancer, brain tumors, and sarcomas. Because it follows an autosomal dominant inheritance pattern, only one mutated copy is enough to elevate cancer risk. Individuals with this condition require close monitoring and early intervention. Tumor suppressor genes can be categorized into gatekeepers and caretakers. Gatekeepers, such as RB, p53, and APC, directly regulate cell growth and division. Caretakers, including DNA repair genes, help maintain genetic stability by fixing errors. Both types are essential for preventing cancer. When either category is compromised, cells accumulate mutations and become prone to malignant transformation. Xeroderma pigmentosum is a rare autosomal recessive condition caused by mutations in DNA repair genes. Individuals with XP are extremely sensitive to ultraviolet light because they cannot repair UV-induced DNA damage. Even small amounts of sun exposure can lead to severe skin damage and cancer. The risk of skin cancer in XP patients is estimated to be more than 200 times higher than in the general population. XP highlights the critical role of DNA repair in cancer prevention. In XP, one of 7 possible DNA repair genes may be mutated. These genes normally participate in nucleotide excision repair, which removes UV-induced pyrimidine dimers. Without this function, damaged DNA is left unrepaired, increasing mutation rates. This leads to early-onset skin cancers, often appearing in childhood. Preventing sun exposure and using protective measures are crucial for affected individuals. Hereditary non-polyposis colorectal cancer, or HNPCC, is the most common hereditary colon cancer syndrome. It is caused by inherited mutations in mismatch repair genes like MLH1 and MSH2. These genes correct errors that occur during DNA replication. When they fail, microsatellite instability occurs, leading to mutations that drive cancer. HNPCC also increases the risk for endometrial and other cancers. Hereditary cancer syndromes can follow either dominant or recessive inheritance patterns. Dominant syndromes, like Li-Fraumeni or HNPCC, require only one mutated gene copy to increase risk. Recessive syndromes, like XP, require both gene copies to be mutated. This distinction affects how the conditions are inherited and who in the family may be at risk. Accurate diagnosis and genetic counseling depend on understanding these patterns. Family history plays a vital role in identifying hereditary cancer risk. Individuals with multiple relatives affected by the same or related cancers should consider genetic counseling. Genetic testing can reveal the presence of harmful mutations and help predict cancer risks. Based on results, patients may choose early screening, preventive surgeries, or lifestyle changes. Early awareness is one of the best tools in cancer prevention. Hereditary factors play a critical role in cancer risk for a subset of the population. Tumor suppressor genes and DNA repair genes are essential in protecting the body from cancer. When these genes are inherited in a mutated form, individuals face increased cancer risk, often at earlier ages. Recognizing these patterns allows for timely interventions. Understanding the genetics of cancer paves the way for more personalized and effective healthcare.